It's such fanciful thinking, and there's all this nonsense that comes out, and sometimes Ed, I think I'm a lone voice crying in the wilderness, and people have accused me of lying. So I say this can't work. I'm accused of lying. I'm accused of many more severe things, and I don't get many organic chemists coming around me and saying, "Hey, Jim, you're right," and because you undergo attack if you say these sorts of things. If you speak the truth in science that's against the groupthink of science, you get attacked. So, Ed, be prepared to be attacked, but I guess it doesn't matter as much for you because you're retired anyway. They can't cut your grant funding.
No. I'm living off my wife's grant funding. She's got her own business, okay. So we have that coming in. But I do understand what you're talking about. There is a very strong community that is protecting the status quo. And the thing I found out early on is, if you go to any college or university, the biology faculty always outnumbers the chemistry faculty. So there's more of them than there are of us. And it's just the organic chemists that have the expertise to see the folly in some of these abiogenesis schemes. The inorganic chemists, they're not going to touch it. Mm. Physical chemists might. But the analytical chemists, they won't touch it.
They don't have the ability to really know. It's unless you've worked with molecules and you've seen how rapidly they decompose and what kind of things you have to do to get them to perform what you want them to perform, and the molecules that are life relevant. So we've talked about amino acids. Amino acids is what were made in these, and the problem is they racemize, they decompose, they react very quickly with carbonyl compounds to make imines. Those imines then start getting attacked either by a nucleophile like cyanide, or they eliminate if there's a nearby hydroxyl that can eliminate, and they start decomposing. But let's look at some of the other class of compounds. So what do you think would happen, say, we'll take one of the simplest ones, the lipids that people think are simple. Lipids are composed of a glycerol that is a 1,2,3-trihydroxypropane, and they'll usually have a couple of tails hanging off generally on the 1 and the 2 carbon. They'll have some fatty acid tails. These are esters. From what I know, esters can hydrolyze pretty easily, and so if they ever got near a hydrothermal vent, they're toast very quickly. And then also, if you're going to try to make those chiral, you have to distinguish between the two ends because the glycerol, the 1,2,3-trihydroxypropane, doesn't have a stereogenic center. It's only when you functionalize one end, when one end forms the ester, now you have this. They're enantiotopic. Actually, the two ends are enantiotopic. So you have to selectively get one and not the other because all of these are chiral. And people have looked at these sorts of things. If you don't have them chiral, if you don't have a stereogenic center there, because the middle one is going to be stereogenic, its antipode, its chirality will depend on which one of the two neighbors got functionalized, then you don't get a sufficient lipid bilayer if you don't have these chiral units there. So you have to have this chiral. So these two ends are enantiotopic. I mean, tell us about how in your mind would these ever form in a friendly way where you can make masses of these in order to make lipid bilayers?
How they form is it's a difficult question. They're very easy to take apart. In another project I did, we were looking at wax esters and triglycerides. And, it's the same bond. It's an ester bond. And the wax esters triglycerides, they're generally too long to do gas chromatography on because they weigh too much.
Yes.
So we would take them apart, and that was real easy. 15, 20 minutes with a little acid or a little base, and you take it apart. I had a friend at Mabari that used to make his own diesel fuel by taking, grease, triglycerides, and cooking it up with a little sodium hydroxide.
Right.
And he can make his own diesel fuel that way. So it's real easy to take them apart. To put them together, that is the tricky part.
That's the tricky part.
You've got,
yeah, and
you got to make the ester. If the fatty acids have any other functional group, you might have a reaction in the wrong place.
they compete, right?
And if the fatty acids are unsaturated, which, I forget the word. We used to call them polyunsaturated fatty acids, but
Yes, they could be cis or trans. Generally, we want the cis. We don't want the trans
Yeah.
there. Yeah.
but in the health food industry, they call them something else. The omega-3s, fatty acids. Those kind of things, they're primarily made as triglycerides or diglycerides or phosphoglycerides.
And they're often allylic alcohols.
Yes.
Yeah.
And so there are all kinds of ways that the synthesis can go wrong. Now, when you have an enzyme system and things, an enzyme grabs this part of a molecule.
Now, just so that the audience knows, enzymes come when there's life. You have to first have life to have enzyme systems working. Yeah.
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 Yeah. So your body and all living organisms can make these quite easily.
No problem.
But if you have a graduate student trying to make it in the lab, you got problems.
Right. And just reaching back now to the amino acids, half of the amino acids have active side chains. So in other words, some amino acids will have a carboxylic acid group coming off, and the other ones will have an amine group coming off. Those are indistinguishable chemically from the alpha amino acid and from the carboxylic acid where you want the chemistry to take place. So these end up participating, and those, and if they participate, it's game over. Yes, nature may use those in couplings, but that's always post-translational modification. That's modification later. You can't have that as the stringing backbone of the polypeptide. And everybody who says that they make polypeptides by hot-cold cycles, where they take a very hot surface, 150 degrees, and they'll put it on there and dry it out, and they'll make, say, an oligomer of, say, a 10-mer, they never use the amino acids that'll have a carboxylic acid hanging off, like an aspartic acid or something like that, or an amine hanging off. They always load the system because you can't do it. It can't be done. Do you agree?
Yeah. Yeah. A lot of these so-called next steps after the Miller-Urey experiment, they stack the deck. They pick amino acids that will react only one way to prevent the side chains, the byproducts, the competing reactions. They make sure there's never any sugars around, because that'll go the Maillard reaction. And they tune the other conditions, the pH, the temperature, to what is conducive to what they want.
Yes. They'll go high pH, low pH, and they'll vary it according. It's all loaded, something you wouldn't have on Earth. Yeah. Okay. So we've dealt with the amino acids. We've dealt with the fatty acids making the lipids. Let's look at sugars, which are carbohydrates making the polysaccharides. Tell me about sugars. I'd seen your interview with Bill Dembski, and you talked about the foremost reaction. Then I sent you this recent paper by Krishnamurthy, and so tell me about the making of sugars.
Well, the foremost reaction was used as a plausible pathway where you can make sugars. Now, we have to be careful because sugars come in all sizes and all varieties. If you have an aldehyde and on the next carbon is a hydroxy group, you got a sugar. But it's only 2 carbons long. The ones you need, ribose, that's 5 carbons. And so you've got to add 3 more carbons with hydroxy groups to that first sugar that had 2. Each step, something can go wrong. And, the first time you add to the 2 carbon, you have 2 possibilities for going wrong. Mm-hmm. But then with 3 carbons, you got an aldehyde and 2 hydroxy groups, you have 3 chances to go wrong. And when you get to 4, now you have 4 chances. And so the number of reactions that don't go to where you want to go doubles with each carbon that you add to the sugar. So it's hard to make sugars non-enzymatically.
Yeah. And in the foremost reaction, you get billions of compounds. People don't appreciate that when you have a billion different compounds that all have the same types of groups, they all have hydroxyl groups, they all have an aldehyde group and things like that, that you can't ever do chemistry on that mixture because other things would compete. They think if you can see it by mass spec, it is therefore there and you can use it. You can't use it. You have to have more constrained chemistry. Wouldn't you agree?
Well, you can do lots of chemistry. Very little of it is what you want. I can tell you sort of a story of my early in my career when I was a synthetic organic chemist. I did very well in class. The professor picked me up to do a summer internship with him, and I was very good at following the recipes for making the reagents that we wanted to use. I could do those kind of reactions. But then when it came time to try something new, to do some new chemistry, starting out with pure reagents. So we've eliminated all the side reactions, but only what's going to happen with our reagent and the compound that we want to either oxidize or reduce. And invariably, every one of my reactions were turned into this gooey mess. And the reason I got so good at analytical chemistry was I was trying to find out, well, what went wrong? What did I make instead of what we thought we were going to make? And I would have these very complex mixtures of organic compounds. And I do IR and NMR and, trying to figure out. And that was the beginning of my end as an organic chemist, because I turned out to be a better analytical chemist dealing with the messes I was making than, and all my colleagues that were working on the bench next to me, they were doing different reactions with different compounds. And they get done, they'd have these beautiful crystals. They take an NMR or an IR and they get beautiful spectra, and I had messes. And it was like, it was frustrating. But in the process, I was learning how to use all kinds of analytical instruments.
Right.
Which, when it came time in graduate school, I was set.
Yeah, it served you well. And that's what you developed to look into the hydrothermal vents, to look in the deep sea chemistry. Yeah.
I remember from this paper, there was an NMR spectrum that was purporting to show ribose.
Yes.
And where the peak was supposed to be was a lump. Well, who knows how many compounds were there? But it wasn't the nice, sharp ribose peak. And I'm like, well, what's going on here? Because those are the strong peaks they should be looking for. And I'm thinking to myself, they didn't show up.
The spectrum that was put out by Benner for ribose, I brought that to a carbohydrate chemist. And I said, would you look at this spectrum? And I said, would you call this ribose? He looked at it, he says, this is garbage. This is garbage. You can't do anything off of this. No, that's not just noise. This is trash, and you can't do chemistry. Now, we'll go to the next step, the polymerization of these. So if you take, for example, six glucoses, people have done the calculations because six glucoses, so you'll have these hydroxyl groups on glucose. You have a number of hydroxyl groups here, and then you have this aldehyde. This can form a six-membered ring or a five-membered ring, or it can be an open form, and you have all these different alcohols. So if you take six glucoses, these can be hooked up. People have done the calculations, 1.2 trillion ways, 1.2 trillion ways that you could arrange six glucoses. This is crazy. If you have six A bases, I mean, how many ways can you hook this together? I mean, A. Now you can have, if it's on RNA, you can have the 2' 5' linkage. You can have some branching, but sugars are so hard to deal with. So if you can't hook these up in the right way, you have both the alpha and the beta at the anomeric center, but then you have all the other regioisomers. Making the polymers and all of those polymerization steps, every coupling is a free energy that's positive. So making the polymers thereof, some people have said, are even harder than making the small molecules, and that may well be the case. It doesn't look like we have a solution to the carbohydrate problem, to the sugar problem, does it?
No. You'll make lots of products, but none of them are what you want. Yeah. And if I remember my sugar chemistry correctly, and now I had the class over a half century ago. So the difference between cellulose and amylose, I think it is an alpha-beta linkage.
Exactly. You remember it properly. It's the alpha-beta orientation at the anomeric center that does this.
Our bodies make amylose to store the glucose for use later. We can't digest cellulose.
Right.
We can't eat wood.
People don't eat wood. People don't eat grass. Cows eat grass because they have an enzyme. They have a microbe in their gut that can actually break that linkage for them.
Yeah. And that just tells us how specific the polymerization has to be.
Yes. So people realize this is just something that's either this way or this way. It's just up or down. You get that wrong, you can't eat it.
Yeah. Well, you can eat it. You just not going to do
it. It goes
good.
right through you. You don't get any advantage from it. It doesn't become part of your body.
They call it fiber.
Yeah, it's fiber. So we can't make the amino acids or the polypeptides. We can't make the fats. We can't make the sugars. There's one class left. That's the nucleotides, the DNA and RNA. Now, a nucleotide has to be made up of a sugar. So if we can't solve the sugar problem, we can't solve the DNA, RNA problem, right?
No.
All right. So we can't make any of these things. And people say, oh, well, did you see that paper? They're taking these trimers of nucleotides and they're doing this. I said, well, wait a minute. If you're really telling me this has relevancy to early Earth, where did they get their nucleotide? And in every case, they got their nucleotide. It came from a natural source. It already came from life, from living biological system. So of the four classes of compounds that build us, Ed, you and I have come to agreement that we can't make the small molecules that make them up, nor can we do the polymerization. Because when you do the polymerization, say of RNA, you can have the 3' 5' linkage, which is what you want, or you can have the 2' 5' linkage, which is what you don't want, and you can have branching off that extra hydroxyl, plus the instability, as you were mentioning, that extra hydroxyl just swings around and hits the phosphate of the one lower to it, and the thing falls apart. So we can't make the small molecules of the four classes of compounds. We can't make the polymers that generate these. We're kind of lost on origin of life, aren't we?
Yes. The Miller experiment was a tremendous advance because it was the first time somebody stopped talking and actually went in the lab and tried to do something. And it created a lot of excitement. But it's been 75 years and we haven't gone very far.
I say this all the time, man. I am so glad you said it before I did. You said it before I did, and this is a guy who worked with Stanley Miller. So we can't get these compounds. And what my argument has also been is we may be able to approach the target a little bit more, but what happens is the target moves much further away from us because we learn the complexity of what is in a cell. So, for example, there's something called the interactome. This wasn't known until about, 25 years ago. This interactome, just the non-covalent interactions, the way things are lined up. The interactome just in a simple yeast cell for just protein-protein interaction, that has 10 to the power of 79 billion. That's a 1 with 79 billion zeros after it. That many possible combinations, and only a few of them are going to work. So you learn these sorts of things. The other thing is chiral-induced spin selectivity, which was discovered about 20 years ago. This chiral-induced spin selectivity, which tells us that reaction chemistry has to have the right configuration on the helical structure, on the enantiomeric structure, because you get electron transfers down these chains, and without that, you get too much heat release. And so you couldn't have had life unless you had that first control. So it's not like enantiomer structures could have evolved to have pure enantiomers later. You couldn't have done this with the racemic compounds. It had to be at the start of life. So all of these problems, that's why my argument is every year when we learn more about the complexity of a cell, the target to make the simplest cell has now become harder. So in essence, we're moving further away.
Yeah. The amount of discoveries in biochemistry that have happened since the Miller-Urey experiment, changes the game. It's so much more complex than the target they had back in mind back then. Mm-hmm. If I remember correctly, I think the structure of DNA was announced
right about
the
that
same
time.
year.
Yeah, right about that time.
As the Stanley Miller, 1953. The target that Stanley had when he started his experiment, they didn't even know what DNA was. And that's how juvenile the field of biochemistry was. Mm-hmm. It's grown up a lot.
It's grown up a lot.
And with each new discovery, the target for abiogenesis gets further away.
Gets further away. And when they discovered that structure of DNA, they didn't really understand the code. That came about 3 or 4 years later, where they figured that you had these trimeric units that are defining for a specific amino acid, and that's how the information is now transferred. I mean, it was they knew so little compared to what we know now about a cell, and we continue to learn so much, and you've got to solve every one of these problems. And people say, well, cells were much simpler back then. And so biophysicists have back calculated what is the simplest cell you could possibly have. And you still have 15 different massive structural components that we have no idea how to make. None of them. And so we're so far from being able to solve this thing. It's a big problem, this whole origin of life problem. And for me as a scientist, I could never sit here and say it will never be solved because I don't know what people are going to know in 1,000 years. I don't know. But as far as we know, we're not even close to solving this in our own laboratory. Putting aside origin of life, this is how life formed on an early Earth. Just saying, can we even take life? We can take a cell, as you know. We can take a cell, deconstruct it, and have all the different polymers there. Have the DNA there, so we already got our code. Have all of that code and everything. We can have all the carbohydrates in their polymerized form as the polysaccharides. Once it's apart, can anybody take those parts and put them back together into a living cell, Ed?
You can't put Humpty Dumpty back together again.
You can't do it. And it's not even that we can't. We wouldn't even know how to go about doing it. You put all these things together in a liposome, they don't all start functioning. It's like taking all the parts of your engine and throwing it inside throwing it on the driver's seat and saying, hey, if I shake up this car enough, it's going to start going. It doesn't go. And so we have no idea. So let alone how it happened on an early Earth, we can't even do it in our modern laboratories. This is how clueless we are on origin of life. Before I wrap this thing up, Ed, I want to ask you a question because I've heard about your background. Do you believe in the physical resurrection of Jesus Christ?
I do.
Good. So do I. And this is what I want young people to see. This whole thing of the resurrection of Jesus Christ is based upon historical evidence that man has risen from the dead. People can have very strong and productive scientific careers and still be believers in Jesus Christ and walking with him as Lord and Savior. And this is what I want people to be able to see. Ed, I want you to wrap this whole thing up for us. Is there anything that you want to say to on origin of life or on faith or any matter you want to talk about?
One of each. Origin of life. I once asked Stanley Miller how long he thought the prebiotic soup was on Earth. You've got an atmosphere of reactive gases. It's reducing. There's lightning. How long did that situation last? And, because that sets the limit for how long you have to make the first living organism, or puts one limit on it. And what he said totally surprised me and caught me off guard. He said 10 to 50,000 years. You don't have 100,000. You don't, maybe 100,000, but you don't have 1 million. And 1 billion years is totally out of the question. You've got a very limited time for where you've got the prebiotic soup and you've got all this work to do. And every time they make a new biochemical discovery, you got more work to do. So that's what I would say.
That's good.
Marginally.
That's good. Yeah.
So
time. We don't have the time that people think we have.
You don't have the time.
Yeah.
And time doesn't help you. Because you and I both know if you're doing a synthesis and it doesn't work, you don't come back the next day, set up the same experiment and say, I'll give it a month, and come back and expect to get a better result.
Right.
The scientific revolution began with the astronomical revolution. Copernicus, Galileo, Kepler,
Right.
Newton. They were all Christians. They all believed in the physical resurrection of Jesus Christ.
Yes.
They did pretty well in terms of science. I think we're in good company.
We are in good company. And the reason people have gone back and they said, why did this come from this Judeo-Christian background? And the idea is that we expected God to work on regular and consistent means. It's not like Poseidon is upset today and therefore the winds are going to come. We don't have a capricious God. We have a God that is reliable. We have a God that is sound. And so we can expect that what we do today, if we duplicate it, it's going to be the same tomorrow and the next day and the next day. You didn't have this in other faiths. You didn't have this in other considerations. And here you have this. And so there was this expectation that we have a God that is regular in the sense that there's consistency. We have physical laws that are consistent. And that was the origin of all of this. Well, thank you so much, my friend. For me, this has been really exciting because I feel like I have touched history. And so for me,
Are you saying I'm an old guy?
well, that you interacted with Stanley Miller of the Miller-Urey experiment, which was 75 years ago, was this experiment, and that you had worked with him and talked with him and interacted with him, and now I get to interact with you. I mean, this to me is like sort of like these Erdish numbers of what's the distance from these Genesis points. But for me, it's a very big deal. Thank you so much for coming on this channel. I know you don't normally do these sorts of things, and so for me, it was great having you. And I suspect there's going to be other people that are going to come knocking on your door and want to get an interview with you because they're going to want to touch history as well.
Well, thank you very much. This has been a lot more fun than, well, this is my first web interview. So I had no idea what to expect, how it was going to go. And it's going very well. I've enjoyed it. Thank you very much for inviting me. And hopefully we've shed some light.